Okay, we'll get started. Welcome, everyone. Thank you for joining. My name is Christophe Diaz, and I'm the Vice President of Scientific Affairs at CurePSP. So fourth as the scientist speaker, Dr. Soto. Before we get started, remember that you're automatically muted and that your video is disabled as you join the webinar platform. During Dr. Soto's presentation, please use the Q&A function to type your questions, and we'll do our best to address them at the end of the presentation. The session will be recorded and uploaded on our YouTube channel, and we'll also email the presentation to those that registered. Please visit www.psp.org for more information. Our speaker today is Dr. Claudio Soto, Professor of Neurology and Director of the Mitchell Center of Alzheimer's Research and Related Brain Disorder at the University of Texas Medical School in Houston. Dr. Soto is also the founder and the Chief Scientific Officer at AMPRION, a biotechnology company focusing on the developing technologies necessary for early diagnosis in Alzheimer's disease, Parkinson's disease, and related proteinopathy. His work and lab has been funded by multiple sources, including the National Institute of Health, the Department of Defense, the Michael J. Fox Foundation, and the Alzheimer Association, and many other. And Dr. Soto will have a slide at the end to showcase the work and the funding sources. On behalf of CurePSP, I'd like to thank our thousands of donors, volunteers, and supporters. All that we do at CurePSP, including this new webinar series and the research we fund, is made possible by the hundreds of donations we receive each month. Today, I would like to particularly recognize and thank Karen Kinzel for her donation to our Ask the Scientist program. Karen Kinzel is the president of the Patrick Risha CTE Awareness Foundation. More information can be found at stopcte.org. Thank you all for your continued support, especially in these very difficult times. Dr. Soto, thank you for sharing your time and expertise with us today, and looking forward to your presentation, and the floor is yours. So if you want to share your slides and get started, thank you again.
Many thanks, Christophe. Do you hear me well?
Yeah, the sound is perfect, and I can see your slides. So
okay,
looking forward to it.
Ready. So thank you for a kind introduction, and thanks for inviting me. It's it's always a pleasure to talk to people, especially with the foundations that are really increasing the awareness and funding and fight for all these diseases that are devastating, particularly PSP. It's a very dramatic disease. I don't work directly in PSP, but many of the things that we do are related. Because as I will tell you at the beginning, the presentation will be mostly in two parts. So the first part will be more to talk about an event that we think is common for all neurodegenerative diseases, including PSP, including Alzheimer's, Parkinson's, multiple system atrophy, and others, which is at the center of how these diseases occur, and how we have used this knowledge to develop novel strategies for early diagnosis. I am convinced that early diagnosis is a main issue in the field, which probably is partially responsible for many of the failures in clinical trials, because the problem with many of these diseases, talking about neurodegenerative diseases, is that they are diagnosed today by clinical examination, and when the clinical symptoms of the disease, the brain the brain is already largely destroyed. So it makes any treatment very difficult to produce a meaningful benefit. So picking up the disease before the disease actually appears, it probably would help substantially treating these dramatic diseases. So I want to tell you the second part, as I said, our effort to develop technologies for very efficient and specific and sensitive detection of what we think is a key event in the disease. So, okay. So first, I would like just to, disclose that I have, as Christophe mentioned at the beginning, I have conflict of interest, and from the point of view, and beside of my role as a professor at University of Texas, I founded and I'm currently the chief scientific officer and member of the board of directors of a small company called AMPRION, who is trying to develop the technology that I'm going to show to you today, call it PMCA or RT-QuIC, for the detection of misfolded protein aggregates in various diseases. So, so let me go from the, the more general. Just tell you, I mean, neurodegenerative diseases are some of the most dramatic diseases of our time. They're chronic, progressive, and they just keep going and going, more and more severe until basically the people die. So they're characterized by selective loss of neurons in various different areas of the brain, depending on the disease. So if the disease is more like a dementia, then you will have more death of neurons in areas related to cognition, memory, etc. If the disease is more related to motor alterations like Parkinson, PSP, or others, you will see more death of neurons in those areas of the brain. So the dramatic thing with the neurodegenerative diseases is that they severely affect the qualities that make human beings special. So including memory, cognition, language, personality, skill, movement. As much as cancer and cardiovascular diseases are fatal and very difficult diseases, they actually don't rob people of their own basic qualities. So when you see your familiar members losing the ability to recognize you, or all their personality, all the ability to move, to walk, etc., it's really very sad. So the biggest problem is that today where there is no cure for any of these diseases. There is some palliative treatment for some of them, but produce only a minor improvement, and it's only temporary. So the diseases can be sporadic and or inherited. So usually each disease of the group has a sporadic, has a hereditary component most of the time, with the exception of Huntington disease. It's a minor group of patients. The majority usually are sporadic, so people who do not have clear mutations that are inherited from parent to child. So in this group of neurodegenerative diseases, we're going to include Alzheimer's, Parkinson, Huntington disease, frontotemporal dementia, amyotrophic lateral sclerosis, progressive supranuclear palsy, corticovascular degeneration, multiple system atrophy, dementia with Lewy bodies, cerebral ataxias, and prion diseases. Many others that I did not mention here, but I mean, no, these are some of the most common. So, as I said, there is no cure, and also there is no early diagnosis to detect the disease before the clinical symptoms appear. So a key event that happened in all these diseases, and this is a commonality that really has to do with the intrinsic nature of what goes wrong in the brain in these diseases, is the accumulation of these misfolded protein aggregates. So here are some pictures of aggregates that are seen in some of these major neurodegenerative diseases like Alzheimer's, Parkinson, Huntington, ALS, prion diseases. You could put any other of the group that I mentioned before. And they all show this formation of these protein aggregates. The main difference between the different diseases is what protein is the component of the protein aggregate. But the underlying mechanism is the same. So it's a normal protein, a protein that we all have, plays probably an important function. Under certain circumstances, and these are usually related to aging, the protein starts to change its shape and become aggregated and accumulate in the brain, forming this deposit that we can see in the brain of people with the disease. So there are a lot of clear evidences that the formation of these misfolded protein aggregates is a key event and the causative event of the disease. For example, and I'm not going to talk about any disease in general. This is a more, any disease in specific. This is a more general slide to say that in general, we see that mutations in the gene of the protein undergoing misfolding and aggregation result in inherited form of the disease. So just changing or making abnormal this gene that encode that protein that end up forming the aggregate produce the disease hereditary. So that by itself tells you a lot, that this process is probably very key. People have been able to make mice models for many of these diseases by simply overexpressing the mutant form of the human gene that encode this protein. And then these mice usually develop the protein aggregates, but also they develop concomitant many of the properties and characteristics of the disease, including in many cases, clear clinical symptoms of the disease. So the protein aggregates are the typical signatures of the disease. Indeed, all these diseases, even though they are diagnosed clinically by presence of the clinical symptoms, the final diagnosis is usually post-mortem when people die and you look at the brain. And for each disease to be diagnosed as such, you have to have the presence of these protein aggregates. It's been shown that misfolded protein aggregates are highly toxic. If you give them to cells in culture, neurons in culture, they will kill the neurons by apoptosis. And one evidence that initially was only circumscribed to a small group of the diseases called prion diseases, but now is clearly seen in many of the others, is that the misfolded and aggregated protein is actually able to propagate the disease in an almost like an infectious manner. I will talk a bit more about this, but this is a key evidence. In prion diseases, which are infectious diseases, but it's also neurodegenerative disease, you actually can take this purified protein, inject into an animal, and the animal will develop the disease. So that again tells you how key is this process. So all our work, and actually the work of most of the people in the field, is based on this idea that the central event in these neurodegenerative diseases is the an aggregation of an accumulation in the brain of these abnormal proteins. There are 4 proteins that are the most commonly aggregated in the human brain. And again, these are associated to different diseases. So among these 4 common proteins are amyloid beta, which is present in Alzheimer's disease, the tau protein, which is also present in Alzheimer's disease, but is also present in several other diseases called as a general Tauopathies. And these include diseases like frontotemporal dementia, progressive supranuclear palsy, corticobasal degeneration, chronic traumatic encephalopathy, Pick's disease, etc. Then the third protein is called alpha-synuclein. And this is a protein that is first linked to Parkinson's disease, but we know also now there are other forms of synucleinopathies that include dementia with Lewy bodies, multiple system atrophy, and Parkinson's disease dementia. And finally, the TDP-43, which is a protein that has been linked to amyotrophic lateral sclerosis and some form of frontotemporal dementia. There is also a new disease that we recognize called late or limbic predominant age-related TDP-43 encephalopathy. So these are besides of these 4 proteins, there are others that are accumulating some of the more rare form of the disease, like Huntington, or the prion protein in prion diseases, or SOD1 in some form of ALS, or ataxin protein in cerebral ataxias. So, but also what happened, and it's very clear in many of these diseases, is that often, even though I say in a simplistic way that, for example, amyloid beta and tau are the proteins that accumulate in the brain of Alzheimer's disease. If you look at the patients with Alzheimer's disease, you see in quite a big percentage of them also accumulations of alpha-synuclein and TDP-43. So it's not so clear-cut as one protein in each disease. It's really, these 4 proteins have, for some reasons, a very high tendency to aggregate in the brain, and they can be present in many different diseases. So, which makes, again, the treatment very difficult, because if you had an agent that is specifically for tau, it may not be enough. You may need to have synuclein, you may need to have a beta, etc. So, okay. So, as I said, I mean, even though there's these 4 proteins, there is 2 proteins that are associated to several diseases. So one is tau. These are the so-called Tauopathies. And Tauopathies include the diseases like Alzheimer's, frontotemporal dementia, corticobasal degeneration, progressive supranuclear palsy, Pick's disease, chronic traumatic encephalopathy. There is a couple of others also more rare. But so what is thought that happens in these diseases is that the tau protein aggregates and accumulates in the brain of these patients. But the reason why the people develop different symptoms and different diseases is because the structure of the aggregates in each of the different diseases is different. even though this protein is the same sequence, you may get aggregates that have a different structure that accumulate in the brain, affect different type of neurons, and that's why produce different type of clinical symptoms. The clinical symptom can be quite different, actually. You know, tau in Alzheimer's disease will produce, you know, more dementia, but the tau in some of the others will produce more motor abnormalities. So, and in CTE, chronic traumatic encephalopathy, is more like a psychiatric disease. So the same happened with the synucleinopathies. So disease is caused by the accumulation of alpha-synuclein. So there is at least 3. So Parkinson's disease, MSA, multiple system atrophy, and DLB, dementia with Lewy bodies. And again, the thinking is that synuclein can adopt different structures that are responsible for the different disease manifestations. So based on what I have said, and the evidence is that the misfolding aggregation of protein is a key event in the disease, we think that the disease pathway that leads to this disease is involved the misfolding of the protein. In this case, I put disease pathways for synucleinopathies. So alpha-synuclein, normal protein become misfolded to form these small oligomers that then grow to form these bigger fibrillar aggregates that are the ones that are deposited in the tissue, in the brain. In the case of Parkinson's disease as Lewy bodies, that leads to neurodegeneration, so neuronal cell death, synaptic alterations, which finally leads to brain malfunctioning, and this finally leads to Parkinson's disease. So the thing is that what we know from many of these diseases is that, you know, the manifestation, the clinical manifestation of the disease can occur at some age, but then all the rest of the processes can occur years, if not decades before. So therefore, if you want to advance in a little bit, what will be the second part of my talk, if you want to come out with a way to diagnose early which people are on the way to develop the disease before they have any destruction of neurons in the brain, you need to really aim for detecting some of these earlier events in the process. So some of these earlier events, like the formation of these misfolded oligomers, and we think that these materials may be actually circulating in biological fluids. We'll talk more about that in a minute. So let's show this one. So how the mechanism of protein misfolding? So how the protein misfold and aggregate? So it turns out that they all fold a mechanism that we call seeding nucleation polymerization process. So this is a process in which the normal protein is the folded protein. It's under certain conditions start to produce some misfolding events, which can be triggered by a variety of different factors, which then become stabilized by protein-protein interaction, forming these oligomeric small seeds that that are able to capture more and more of the normal protein and grow in an exponential manner to form these fibrillar aggregates that are deposited in the tissue. In fact, if you do like in a study in the lab, in the test tube, and you put the protein under certain conditions, you will see that at the beginning, and you measure aggregation over time, you see at the beginning nothing is happening, because all what is happening is these early misfolding events. But as soon as you get the formation of these first oligomeric seeds, the whole process becomes accelerated, and then you see this rapid aggregation. In fact, if you add these oligomers that are forming in a different reaction, you can speed up the whole process. And this is the key event that we will use for our strategy for diagnosis, and also is a key step that happened in I was telling you before that prion disease is one of these more rare form of the neurodegenerative diseases. They are infectious disease, and the way that they're infectious is because the abnormal prion protein add as a seed that can trigger and speed up the process of the disease. So, so this is what we call, I will call the prion principle. And the reason I talked about this is not because it's important in this small group of diseases, but because recent research by us and many other investigators have shown that this principle is at the root of all neurodegenerative diseases. So this process of seeding nucleation I was showing you in the previous slide is actually common for all these different diseases. So being A beta, tau, alpha-synuclein, TDP43, whatever protein it is, it grows in the same mechanism. So they all have the potential to be speed up by adding preformed aggregates. So this is what the prion principle posits, that the biological information can be faithfully transmitted. Basically, biological information in terms of protein aggregation can be transmitted and self-perpetuated by an autocatalytic propagation of this abnormal protein conformation in the absence of genetic changes. This is basically what we call the prion hypothesis that was awarded the Nobel Prize to Stanley Prusiner to show that this is a new form of infectious agent that can produce disease composed exclusively by a protein. So the thing is that the prion principle, we think, is at the root of the most prevalent and incurable diseases of our time, including Alzheimer's, Parkinson, and not only, I'm not going to talk about this, but we are doing some work on this area, not only in brain diseases, but also in some systemic disorders like type 2 diabetes and cancer. So what the prion principle says is that when you have the formation of this small amount of oligomers, they can actually take up the normal protein and grow and make it grow in a cyclical manner to get more and more of this protein, toxic protein. When these levels of this toxic protein becomes too high, then the people will go into the disease. So, but this process can go for decades in a silent manner until, you know, enough material, toxic material is accumulated to produce damage of the neurons and disease. So, so prions have the ability to move. And this is one thing that is probably why the prion principle is so important in the other neurodegenerative diseases that have not shown to be infectious. Because just to clarify, only prion diseases are known to be transmissible from human to human. But these other diseases, we and others have shown that you can actually accelerate the disease process by adding these misfolded aggregates into the animal models or cellular models. But what is shown also is that some questions. I don't know if you want me to answer questions now or later.
No, I'll take care of the question towards the end. We'll have like around 10 minutes, and I'll deal with that, so you don't have to be distracted by that. Carry on. Thank you.
Good. So in diseases like Alzheimer's or Parkinson, like the tau protein, for example, the alpha-synuclein protein, what we see typically is that the whole problem starts in one area of the brain and then start moving with time to cover basically the entire brain. And it moves from cell to cell. And what it moves is this. So the formation in one cell, perhaps the process will start, form these aggregates, and they will actually move to the adjacent cells to propagate this process. So that's why it's a very important process in the disease. And we know that the prion principle operates at different levels in different diseases. So like in prion diseases, you know, they're transmissible. You can be transmitted from human to human or animal to human. But in the other cases, there is a molecular event of transmission, but also the cellular transmission between cells and between different areas of the brain. So, so basically the basic of the prion principle of this disease propagation is that the normal protein becomes misfolded, forming these small oligomers that then grow and then at some point break down to release more of these units, and then more and more of this process is happening to end up like in a funnel that you keep going and going and going and going until you have huge amount of these toxic proteins in the brain. So this is what I think is at the center of all these diseases. So what we have done many years ago is to actually try to reproduce this principle in the lab for as a way to study the disease, but also as a way for high efficiency detection of the process very early on during the disease, much before any clinical symptoms or brain damage has ever occurred. So we published this article in Nature initially focusing on mimicking the process in the disease that is the most well accepted disease that involves this mechanism, which is prion diseases. So we basically, what we wanted to do is to use the ability of these abnormal oligomeric misfolded proteins to convert the normal protein in this slow but cyclical process until and this is what happened in the disease, that you know, when an animal is infected with these prion oligomers, then it takes a long time until enough of the abnormal protein is accumulated to produce disease. So this can take, we know because this is an infectious disease, we know we infect, we know when infected, we know when the disease appears. In rodent models can take months, in mammals, larger mammals can take years, and in humans can take up to decades. So and you can see that easily if this is the situation you have where you have very few, let's say one particle of the abnormal protein, being prions or alpha-synuclein or tau or whatever, in the system, let's say in a given biological fluid, let's say cerebrospinal fluid or blood, and you want to actually detect it, it's too little to be detected. So what we do is, let's do what happened in the brain for detection. So getting huge amount of the protein, and then will be easy for anyone to detect. And this is what we do, and we created this technology we call protein misfolding cyclic amplification. It's also well known in the field as RT-QuIC, but don't get confused. It's basically the same idea. It's just depending on how it's slightly different experimental condition you use to do the process. But so what is behind is that the whole idea is that we want to try to detect this small amount of oligomeric seeds by their ability to grow at the expense of the normal protein. So we add the normal protein here in green circles to the little amount of these seeds. Then what happens if we let them incubate, that these seeds will grow by recruiting the normal protein and inducing it to misfold to be able to bind to and become part of the aggregates. So what happens is the aggregate become bigger. So this process, you can see, it may take a long time, but the way that we accelerated this for facilitating detection in the laboratory is that we add a step in which we fragment these aggregates. Once they have grown for some time, we come, let's say, with like some kind of scissors. We use sonication or checking to do that, to break down these big polymers into smaller pieces, and then we keep incubating with the normal protein. Now what we have after this is that it's that each of these units is growing now simultaneously at the expense of the normal protein, and when they have grown enough, then we cut them again into many smaller pieces, and we let them again grow at the expense of the normal protein, and then we cut them again, and we let them grow again, etc. So this is a cyclical process. You can do as much as you want to detect very tiny amount of the abnormal protein. So it's, in a way, is very similar to what we do when we do PCR amplification of DNA. In this case, we are doing amplification of misfolded proteins. So, okay, so that's the technology. We have used this technology for multiple applications, like in the prion field, for example, to understand the biology of the prion disease, develop detection system, to identify treatments, to assist in implementation of regulatory decision, etc. So these are some of the, I don't really have time, I'm not going to talk about prions at all. Just to show you that we have published a number of articles over the last years to show that we can, for example, for the first time, generate infectious prions in the laboratory using this technology, which was considered to be the final proof of this hypothesis that the only component of the infectious agent in prion diseases was just the misfolded protein aggregates, and many other experiments. But what I'm going to focus more in the rest of the talk, it will be more the use of this technology for sensitive detection. And first, of course, we try with prions, and then we publish a number of papers showing that we can actually detect very tiny amount of prions in biological fluids of patients with the disease, both in blood and in urine. In fact, we actually estimated that we can detect as little as one oligomer. So one oligomer is enough to start the reaction, and the reaction is very specific. It's now being used currently in Europe and in the US for facilitating and confirming diagnosis of these diseases. So, but prion diseases are rare. So what we decided to do was go into the more common neurodegenerative diseases that are also the prion principle is also key event. Of course, composed by a different protein with different characteristics, but the process again is the same. So we say we should be able to optimize this technology for detection of protein oligomers associated to all these different diseases. And we published early on an article using the amyloid beta protein in Alzheimer's disease. And more recently, we have published several articles showing the use of this technology for detecting the alpha-synuclein protein in Parkinson's disease and related synucleinopathies. So today I want to talk more, focus the rest of the discussion in our work on synuclein, and particularly I'm going to talk about multiple system atrophy, because I think you guys have a lot of interest in that. It's a disease that have some similarities and clinically, I think, with PSP. And so I will show you some data to say that we can detect these proteins, but also we can actually distinguish the proteins by the structure of a protein. We can actually tell which disease the people have or will have. So let me tell you a bit more. So I told you again, this is the same principle as I told you before. So we use the ability of these oligomers that might be circulating in biological fluids of patients for patients to be. And then we use this ability of these oligomers to seed the misfolding and aggregation of the normal synuclein protein to be able to detect. So this is the first paper we published in JAMA Neurology, showing that we did with the Parkinson's disease, DLB, and MSA, showing that we can actually discriminate very well CSF from patients and controls. So here I show you one of the results of this paper, where we show each dot is one different Parkinson patient, and the red dots and the black dots are, each dot is a different healthy control. So you can see that most of the Parkinson patients have a very high, very clear signal, whereas most of the control have no signal whatsoever, except for these four that gives us a signal in the range of what you see in the patient. So one thing that I need to stress out is that clinical diagnosis is not 100% accurate. So clinicians, even though even the best clinicians, they can never get the 100% sensitivity and specificity on the disease diagnosis, because the symptoms are not so clear also either, dependent on a lot of factors. So what happened in this case is that these two patients, at least, that we were given to us blindly as controls, our colleagues actually, because they follow up these people, they show that several years after that they donated this sample CSF, this was running CSF, cerebrospinal fluid, these people have actually developed Parkinson's disease. So that was very interesting, because it suggests that technology can actually detect preclinically. So, and this is the type of sensitivity we got in this study, 88.5 for Parkinson, 100% for DLB, 80% for MSA, and with a specificity in the range of 97%. So we initiated many studies, and this was one done sponsored by the Michael J. Fox Foundation. What they did is to do the study in three different sites, three different investigators. They gave the same samples. These are very well collected and validated samples over many years, again, sponsored by the Michael J. Fox Foundation. They give it to us as well as to other groups that run the same technology with small variations. And we basically, this is our result. We got again very clear differentiation in the signal between, you know, the Parkinson patients versus controls. And we got 95% sensitivity and 90% specificity. And another group done in UK, Alison Green, got very much similar results. And most of the false positive and false negative were the same in the two studies. Then later on, a third group did the same, Byron Powey, and got basically the same results. So this was all done blindly. So this is a good validation that the technology indeed work and can actually detect with very high sensitivity and specificity the disease in already existing patients. So one of the things that we did and we noticed in very early on when we were doing these studies is that when we get samples of two very similar, clinically similar synucleinopathies, Parkinson's disease and multiple system atrophy, when we get given samples of those, they look quite different actually in our assay. Even though they were both positive, they look different. So here I show you, for example, some of the graph that we got. So this is aggregation. Fluorescence is what we use to measure the aggregation of the protein. Again, remember, we're aggregating the normal protein that we add, seeded by the oligomers present in the seeds present in the biological fluid, in CSF. So what we monitor is the aggregation process. So if there is no seeds, if these are healthy control, you see nothing. There is no aggregation whatsoever. And when you have patients with multiple system atrophy, we saw that most of these patients actually, all these blue lines here, they're all individual patients. They all run into a similar range. Whereas when we did Parkinson's disease patients, these are the red lines, most of them run as in a different way. So the maximum signal you can see is substantially higher than the maximum signal that you see for the majority of the MSA patients. So, these 3 or 4 red lines are patients that were given to us as MSA, but we think that actually Parkinson, because they have the signature of Parkinson's disease. And again, we have 3 here that they were given to us as Parkinson, but we think they're actually MSA because they have the signature of MSA. So why do we see these differences? Well, what we're using here, it's a dye that specifically bind to this fibrillar aggregates that we produce in the reaction. And what is very interesting is that this dye can enter in a different way depending of the structure of the aggregate. It could actually enter more or less, which will give a higher or lower signal. This is where we think this gives us the first idea that Parkinson's disease and multiple system atrophy are associated to different structures of alpha-synuclein aggregates. So we went on and characterized, so we published this actually this year in an article in Nature. So we went on and characterized what was happening here. So the first thing that we wanted to make sure, just to control, because another explanation for these differences between the maximum fluorescence is that for some reasons, all the protein was aggregating in Parkinson, but not all the protein was aggregating in MSA. So we measure the amount of protein aggregated in both diseases, and you can see, and this is the amount of protein that was aggregated measured by a different way. In this case, we centrifuge it, and considering that the size of the aggregate is much bigger than the normal monomeric protein, so we can separate it, and we can see that the MSA and PD, they have really the same amount of aggregated protein. So the amount of aggregate was not different. So what was different? We saw that this again is a good indication that what we are seeing is a different structure of aggregate. So moving to other techniques, what we did was to say, well, this is in CSF of patients. Do we see something similar in the brain? So what we did was to take brain homogenate, in this case for 3, so we took the brain from a person who had died with the disease, we did an homogenization, and then we used the brain actually assess it for our reaction. And you can see that again, we saw the same difference between Parkinson's disease and MSA. And this data is proof that, that what we see in the CSF is representative to what is present in the brain of this patient. So that's very interesting, very important. So the other thing that we did was to, study what was going on. Why were the difference between Parkinson and MSA alpha-synuclein strains? So we used some other fluorescent dyes that have shown by other people to differentiate between different shapes of aggregates. And here I show you two of them. So one, these are the studies in CSF. So this is one dye that we just screened it several, and we found that this one was, because of the structure of the synuclein aggregates in in Parkinson, it binds very well, whereas it doesn't bind at all to the aggregates in multiple system atrophy. So, and this is in CSF, this is in brain. So in brain, we got exactly the same, that the dye binds to the Parkinson, but not to the MSA. Then we got a different dye that it seemed to have a little bit higher recognition for MSA than for Parkinson. It was not so, striking as the one that we found in Parkinson. But again, in CSF and brain was the same. This was what we published in the paper. We continue this screening, and now we have another dye that I did at the time of the publication we didn't have. Is a dye that is only MSA. So now we can only detect alpha-synuclein aggregates that have the structure of MSA aggregates. So you can see that Parkinson is really silent. What was very interesting is that we used this dye for looking at in the brain. So here is the brain of Parkinson and MSA patients, both staying with the anti-synuclein antibody. And you can see the typical Lewy bodies inside neurons. And in the you see these aggregates that are present in other cells like oligodendrocyte mostly, and they're very morphologically different to the one in Parkinson. What was interesting is that when we used this dye that is specific for MSA, the picture in the Parkinson was completely black, so no signal. Whereas in MSA, we have all this labeling, again, suggesting that this dye is specific for MSA. So this is very interesting because we can now develop this dye, for example, for, we are trying to do that for imaging, as an imaging reagent to have an imaging system that you can actually look for MSA specific aggregates, for example, for differentiation between Parkinson and MSA. But for us, it tells us that the aggregates are probably structurally different. So the other thing that we did was to do limited protease resistance. One thing I didn't say and say now is that, one of the differences between the normal protein being A beta, tau, synuclein, prions, and the abnormal form of the same protein is that normally the abnormal form of the protein become highly resistant to degradation by proteases. So most of the proteins are very easily degraded by proteases, but since these are aggregates, they're much more resistant. So we did a limited protease resistance using proteinase K, one of the strongest proteases. And again, in many patients with PD or MSA, and we found that this different using different antibodies. I'm going to concentrate on just this one in here, that you can see 5 different patients of Parkinson's disease and 5 different patients of MSA. They have a very different signature, which means is that the accessibility of the protease to the aggregate is different in Parkinson and MSA synuclein aggregates. So that, again, we reproduce the same result. This from CSF, and this is from brain, again, showing that what we see in CSF is representative of what we see in the brain. So this technology allows us to do a lot of things. So one of the things that we can do is to generate pure structures from Parkinson, from MSA. We can do this serial replication. We can generate large amount of material for structural studies. So here we do several rounds of to generate more and more of these abnormal proteins. And we use this material for structural studies. So we did some studies that are done to measure the secondary structure of proteins. Why? Because the big difference between the normal protein and the misfolded protein is that the abnormal protein, the abnormal protein is very rich on a structure that we call it beta sheet rich structure. So this is a way that the aggregates form by forming this beta sheet rich structure. And we measure this by these two techniques, circular dichroism and Fourier transform infrared spectroscopy. So both techniques show us that what we were having there is the MSA aggregates had a much higher beta sheet structure than the PD aggregates. And finally, we did a technology that is very popular these days. Is a recently used technology to measure, to study the structure of proteins. This can be used actually to atomic detail, so you can have the atomic resolution structure of a protein aggregate. And this has actually been done already for synuclein. So what we did was to compare our MSA and PD aggregates coming from the brain, from the CSF of patients, and we saw that they're actually very different. So this is a model of how the aggregates will be for Parkinson, and this is a model how the aggregates will be for MSA. You can see that the twisting rate is much more frequent for MSA than it is for Parkinson. So we're now working on determining the atomic resolution structure of these two different structures responsible for Parkinson and MSA. So what is fascinating is that these different structures have different properties. So in one case, in Parkinson, they will accumulate in neurons and they will attack neurons. And in MSA, they will actually attack different cell type in the brain called oligodendrocyte and will accumulate in those cells. So the other thing that we found is that these aggregates are also biologically different. So in this case, what we did was to measure toxicity. So how toxic are these aggregates to neurons in culture? So we put the Parkinson or the MSA aggregates in culture, and you can see that the MSA aggregates are much more toxic. It reduced cell viability substantially compared to the aggregates in Parkinson's disease. We have much more data, but really don't have time to show you, but that we have been using cellular models to propagate these diseases, and we can actually propagate it also in cells, these different structures. So just to finalize, I would like to say that so this is one example. What we are trying to develop, we're working very hard on developing our assay also for detecting the other proteins, particularly for you guys, you'll be interested on the detection of tau aggregates, because this is the main protein aggregate involved in PSP. And, but also, of course, for you, it's also important to distinguish from other diseases that have similar clinical manifestation like MSA. But we are developing the tau assay, and we hope to be able to, again, differentiate the aggregates associated to different tauopathies, Alzheimer's, PSP, CVD, and FTD, etc. Okay, so what we think this technology has many applications. So we can increase sensitivity of clinical diagnosis. So we use in patient diagnosis. One thing that I didn't say, but we have already data, is that we can actually distinguish idiopathic form of Parkinson's disease from other forms that are not really produced by damage of neurons in the brain. We can use it to monitor disease progression. We have also some data on this. So we're running a very large study, again, sponsored by the Maggie J. Fox Foundation, that where samples were taken from patients at different stages of the disease, so that we can use it to see if the amount of aggregates that we can see in our reaction correlates with the severity of the disease. So this will be very, if that works, will be very useful to monitor disease progression in patients, but also for clinical trials. So the other thing, as I said, we focus a lot in this today, is we can use this technology to differentiate different forms of synucleinopathies, and hopefully in the future tauopathies. So to help physicians discriminate between Parkinson's disease and MSA, as well as other disease with similar symptoms like PSP. So the other thing that we are doing, putting a lot of effort now, is that we think that there is not just one aggregate in Parkinson's disease, but there are multiple different aggregates, which may correlate with the heterogeneous manifestation of Parkinson's disease. So some people have more motor problems, some other people have more hypokinesia or tremors, etc. So we're trying to correlate the structure that we see in CSF with how the very, very important. So one of our major goals is a preclinical detection. So we would like to get this technology to work early on so that we can actually detect people who are in the way to develop Parkinson or Alzheimer's disease or PSP or Ph.D. or whatever disease, way years, if not decades, before they get the disease. How this will help? Well, you can say, well, how is it going to help because there is no treatment? Well, first it will help development treatments, but also you can actually do some things when the disease is weak. Let's say when you just start to accumulate this aggregate, it might be possible for some lifestyle changes. You could slow down substantially or even stop the progression of the disease if you start very early. So we are doing, again, a large experiment on this, in which what we are doing is to take samples from people with a prodromal form of Parkinson's disease. So there are people who have symptoms of what we call REM sleep behavioral disorder, that people who during sleep move, etc., uncontrollable. So many of these people actually end up going into Parkinson's disease or MSA. So, so what we are doing is to take, some people collect samples from these patients before they went to develop Parkinson's disease and monitor them longitudinally until they develop the disease. So we're gonna try to detect, see which one of these samples we can detect early before the disease appears. We're also collaborating with various companies to develop new therapeutics, because this can serve as a screening assay for drugs. If you have a drug that interferes with alpha-synuclein or tau, spreading and aggregation, you can put it in this system and see if the drug is efficient. The other thing that is very important is for patient enrollment in clinical trials. So many cases, many times the treatment has failed because you actually don't have the patient you think you have. So imagine if you have a drug against PSP, I mean, how many confounding diseases you will have, because there are so many diseases that have similar clinical manifestation. You really need to have a molecular definition that this is a PSP patient versus this is a Parkinson or MSA. So like in the case of PD and MSA, I was telling you, we can clearly distinguish patients with PD and MSA using this this assay. So if you have an MSA specific treatment, we can help you to make sure that the patients have, this disease.
Sorry, I just want to jump in because we have a few questions and some good ones.
I just want to.
Are you okay? That's your last slide, right?
Yes, it's the last. Just,
all right. So I know.
To say that it can be used for, we're trying to go at the end is to some personalized medicines to give the people the right treatment that they need to have depending on the disease they have. So all these different applications, just wanted to acknowledge people in my work. So most of the work here I talk was Sandra Prisco, Mohammad Shanawaz, Abhishek Mukherjee, Nicholas Mendes, and Prakuti Ravadia, although many other people are in the group, and collaborators all around the world, as well as the funding agencies for support. That's it.
Thank you. I cover a lot of material there. So we have a couple of questions around some that you partially addressed. So there's one that asks around if you think the seeding capacity of CSF alpha-synuclein samples, related to the clinical trajectory of the individual Parkinson or MSA patient, you think there will be a proportionality in the differences of the samples as you see now in tau seeding capacity in AD. There's recent study, Dujardin 2020, that show that there is a kind of a profile that is correlated between the severity of the disease and the tau seeding capacity. So are you expecting such a similar correlation between alpha-synuclein and the clinical trajectory in Parkinson or MSA?
Absolutely. I mean, as I said at some point, we're doing a large study to study this in practice, but in theory, and the experiment we have done so far. So in theory, this aggregation process triggered by seeds, it's really should be quantitative, because the more seeds you have, the faster the aggregation will start. So in fact, what we do, for example, take a healthy CSF and add these aggregates that we produce in the laboratory, add different known quantities of these aggregates in the CSF, and we can get patients that, CSF that have very fast aggregation, CSF that has very low aggregation, depending on the amount of seeds. So we can actually have like a calibration curve with the known quantities of aggregates and know more or less what type of, how much aggregates a patient would have. And then a recent experiment we did in collaboration with Maggie folks, we actually already, and even though we have only a small number of samples yet, we're going to run many more, we already have some indications that the technology can differentiate by how rapid the aggregation process start patients with severe or early stages of the disease.
Okay. Thank you. Another one, focusing a bit on the characteristic of what defined the seed. So you mentioned oligomers as the seed. Is this always the case? Can we have monomeric conformers that act as a primary seed?
We don't think the monomers it's able to seed. We do not know, and purposely I was not clear, because I think the field is unclear what constitutes a seed. So we think that oligomers can be seeding, but also fibers can seed. People work with fibers, they sonicate fibers to make shorter fibers, and these are also very, very good seeds. So we really don't know. What we are doing now is to try to characterize the size of these aggregates that are seeding competent in CSF. So the problem is that so far you cannot do this because you cannot see them. But now that we can see them by amplification, what we are doing is to fractionate them. Take the CSF, fractionate it by size, and look at what size range are these seeds present. So far the data indicate that it's something smaller than 2 million Dalton molecular weight, which is already pretty big, but they're certainly not monomers.
One more, do you think that the CSF protein, and if you think of the conformation, the post-translational modification profile that you detect on, let's say, a CSF alpha-synuclein, do you think it's different from what you would detect in the brain at the same time point? Like, what do you think are the differences in the alteration that are CSF specific, and that might maybe take the information away from the direct function that this protein play in the brain?
Yeah, so far the experiment we have done show that what we see in CSF is the same that we see in brain. But we need to do more studies, and that's why we're very interested in identifying these dyes that specifically see one conformation and not the other, so that we can use as little as manipulation as possible from the brain to be able to show that what we see in CSF represent to what we see in the brain or not. So, so far, the data show that what we see in CSF is highly representative to what's present in the brain. One thing that we are, I should say, that I didn't have time to say, but we are actively, and this is where we're putting most of the effort today, is to develop the assay to work in biological fluids that are more amenable for routine use, like blood, or urine, or saliva. So we are working in parallel in all these different fluids to see if we can actually detect misfolded oligomeric aggregates in those biological fluids. It's more difficult because the quantity is lower, and these fluids, especially blood, are much more complex, but we are doing a lot of progress, and we hope to have blood assay like this, hopefully soon.
Okay, we are a couple of minutes past the hour, so I'll just want to thank you, Dr. Soto, for the presentation and putting all this material together, and thank you for our audience. There's a couple of questions we were not able to address, but we'll address them offline and send an email to the group with your recording to follow up the conversation. So thank you again for your work and your time today, Dr. Soto, and thank you all for the audience, and have a great weekend all.